Welcome back. This video is about factors that control infectious disease. So they control how likely it is, how severe it is, how long it lasts, lots of things like that. And then in a different section, we will look at factors that control the spread of disease. So that's epidemiology. So that will be a later video. But for now, we're just looking at all the different factors that change how severe or likely a disease can be. And so this is split into really three different major areas. So virulence is one. How likely is the pathogen to cause disease? How good is it at evading the immune system? That's virulence. And then conditions of contact have to do with what part of the body does it touch, for how long, in what form. And then resistance of the host is how good is the host's immune system. And so these together can be used to predict the likelihood of disease. So we start with conditions of contact, and those are going to be time, dose, and portal of entry, and I'm going to skip through a lot of this. The infectious dose or infective dose, same thing. This is a mystery. Why is there such a thing as infective dose? In some cases, it's hard to know why 10 cells are enough to cause disease, but 1 wouldn't be. That's crazy to me, but that's what we measure. So infective dose is something I talk about in class a lot, and I brought this up, the Unit 2. But again, this is the minimum number of cells required to cause disease. And here is a figure showing estimates of infective dose for different pathogens. And it's a logarithmic scale, so you're looking at the powers of 10 here. So 10 to the 6th power, 10 to the 7th, 10 to the 8th. So Vibrio Cholera is kind of famous because some strains of it have a very high infective dose. And so somebody who gets cholera has to have ingested 1 billion of the cells or 10 billion of the cells. And others are much more effective, and you only need to ingest 1 million. And remember, you could have 1 million cells in crystal clear water. In every milliliter of that crystal clear water, you could have 1 million cells, and they would not affect the optical properties of the water. So it's very hard to avoid ingesting numbers of bacteria at 10 to the 6th or lower. Some of the ones that I point out are some of the nasty kind of gut pathogens, different strains of E. coli that are adapted to cause disease in the gut. And these have very low infective doses. 10 to the 1st power is 10. So there is evidence that this strain, this would be what 0157 H7, if you ever have heard of that. That's this, and 10 cells are enough to get a person sick. The same thing for tuberculosis, Mycobacterium tuberculosis. If you inhale a tiny number of the cells, you get infected, and that's why up to 1/4 of the people in the world have been exposed to Mycobacterium tuberculosis. Here are some important words. Virulence is an important word. This tells you how capable a pathogen is of causing a disease, and we measure it using LD50. So this is a better kind of measure. Instead of a minimum, this is kind of an average. So how many cells are required to kill half of the laboratory animals? And you could also measure infectivity, and that you would measure with the infective dose or the infectious dose. How many pathogens does it take to infect half of your lab animals? Those are important measures. Something that will come up in Unit 4 are virulence factors. These are usually proteins bacteria have that help them cause disease. So they might be a toxin they release into the body, or they might be a signaling molecule they make that confuses the immune system. There are lots of different virulence factors for lots of different pathogens, and each of these contributes to disease. So these are all different examples, if you're curious about different examples. Here's an important one that you've seen before. Endotoxin, or lipopolysaccharide, is a component of the outer membrane of a gram-negative bacterium. That's endotoxin. Exotoxins are secreted or excreted intentionally by bacteria. They're little proteins that get pumped out of the cell intentionally to damage the host. So endotoxin is a fundamental part of the cell. So it doesn't exist to cause disease, but an exotoxin exists to cause disease. We can also think about different things that affect the host's resistance. So you can look at that. These are lots of different things that can change a person's susceptibility to diseases. I don't have much to add to these, so just go with what you read, and I'll see you at the next video.